Is everybody in? Yeah, go ahead and close that. We don't disturb the people who are trying to enjoy the coffee and relax. Okay, so describe the process of photosynthesis. Explain the difference between photosystem 1 and photosystem 2. So the overall process of photosynthesis is what? You're converting carbon dioxide and water using light energy to produce a carbohydrate or sugar and oxygen, right?
So it's converting carbon dioxide?
Carbon dioxide and water. The plants take in carbon dioxide, right? We give them water where it rains. That water and carbon dioxide inside the plant will be converted from that to a carbohydrate or sugar, which when we eat the plant, we get that, right? And oxygen, which the plant puts off while we can breathe, right? Okay, so that is the basis of photosynthesis. Now, it's broken up into two photosystems. There's the photosystem 1, which is light dependent, meaning that it depends on that sunlight or ultraviolet light in your grow room or whatever, right? And then there's photosystem 2, where it is light independent, meaning it does not need light, but yet this conversion of carbon dioxide and water still occurs, producing that carbohydrate and oxygen. So during photosystem 1, what is occurring?
Depends on light.
Hmm?
Depends on light.
Yeah, but what kind of chemical reactions are going on? Okay, you're going to get ATP synthesis, as well as synthesis of what? Electron carrier. What's the name of the electron carrier that is being produced?
The NADH?
The NADPH.
Oh, NAD.
Yeah, when I began this lecture, I talked about the 3 types of electron carriers. That was NAD, NADP, and FAD, right? So now we're talking about NADP.
Sorry.
So when it picks up electrons, it becomes NADPH. And this is occurring during the light dependent reactions. Now this ATP and NADPH that are produced during the light dependent photosystem 1, that's going to be used at night by the plant to carry out its chemical reaction of converting the carbon dioxide and the water into that carbon the carbohydrate and the oxygen, yes.
And that's still part of the light dependent.
This is part of the light independent. So the energy that is being produced from that light dependent reaction is now being used. That's why we can say that photosynthesis is the using of light energy, right? That energy that has been produced during the light dependent portion of it is now being used at night, right? So light independent, you're using ATP and NADPH to produce the carbohydrate and the oxygen. NADPH.
To what?
To
Carry that chemical.
to convert the carbon dioxide and the water into a carbohydrate and oxygen.
Carbon dioxide and water.
Yes.
Into what? Into
Carbohydrate.
sugar
Sugar
and
and oxygen.
Carbohydrate, sugar, and
Carbohydrate is a sugar.
oh, And oxygen?
and oxygen, yes.
So that's photosystem one.
Two.
Photosystem 2 is the light independent. Photosystem 1 is light dependent.
Right.
Would you use the synthesis of the NADPH and ATP of the light dependent one?
Yes,
So that
that's being used at night.
So using ATP and NADPH is for light independent? Or-
Yes. It's being used during the light independent for energy to do this. Everybody good? Hold on. Is that a sausage sandwich? Why didn't you get bacon? I knew I couldn't trust you. Can't trust somebody that don't like bacon. Huh? You know they do have low salt bacon.
Bacon is bad for you.
Huh?
Bacon is bad for you.
It's not.
Yeah. It causes cancer.
Everything causes cancer.
But with bacon,
At least you're gonna be happy when you eat cancer.
I don't like it.
Can't trust you either.
All right, move on.
You like chicharrones? No?
I don't like nothing that comes from.
Huh?
I don't like nothing that comes from pigs.
Oh. So you don't like carnitas either? Ham?
No,
Okay, I tried anything but pigs.
unless it's turkey ham.
That's not pigs.
I know that's why I like it because it's not.
I like turkey bacon.
I like turkey bacon. No,
You eat hot dog?
No, unless it's.
Oh, that's everything.
Nothing.
Okay. All right. Next one. Describe the two types of regulatory mechanisms involved in the integration and regulation of metabolic functions. Now, in your PowerPoint, it just basically gave you a statement for both of them, right? What are the two types of regulations of metabolic functions?
Control gene expression, control metabolic expression.
Okay, so you're controlling gene expression and controlling metabolic expression. How are they integrated? Does the control of gene expression, how does that affect the metabolic expression?
Controls the amount of time in protein production.
So it controls the amount and the timing of protein production or enzyme production. The amount and the timing of enzyme production.
Wait, what?
The gene expression controls the amount and the timing of the enzyme production. So, say for instance, a cell has been placed into an environment that has a high glucose content, right? So, because of that glucose in the environment, that's going to trigger that cell to start producing enzymes that will work on the glucose, okay? And so, if there's a large amount of glucose, the cell is going to produce a large amount of enzymes to work on that glucose. And so that gene will keep on being expressed until it is no longer needed to do so. So when the glucose level has been diminished, then now we have that integration, right? Once that amount has been diminished, the metabolic process will go back until that gene stops producing the enzyme. That's how they are integrated. Kind of like that feedback loop, right? We'll get more into that when we start talking about the lac operon and the trip operon, because that's going to really solidify the explanation of that, okay? But just know right now that we have genetic expression that controls the timing and the amount of the enzymes, right?
Okay.
Okay. The metabolic portion of this, the metabolic regulation means that when a product has been either overproduced or when a substrate is in such low amount that it cannot use it anymore, then it will signal to the genetic portion of this regulation and tell it stop producing the enzyme for glucose, start producing the enzyme for something else, like lactose, okay?
So when
So-
the product is overproduced?
Or when the substrate is in such low amount, it needs to find something else. So if glucose has been used up, but there's lactose there, right? That will signal the DNA or the genome to switch its gene expression. That switching of the gene expression means that it's going to stop producing enzymes for glucose metabolism and start producing enzymes for metabolism of something else, such as lactose, okay?
So will signal DNA to switch?
Yes. So it's integrated.
So when the substrate is low, it signals the genome to switch its gene expression.
Yes.
Okay.
That was pretty simple, right? Now, these are your bonus questions here. And if you haven't noticed the pattern, that the bonus questions are always just the last. So I always get somebody saying, well, which ones are the bonus questions? You should know them all anyways, right? So, describe the lock and key theory of enzyme function. We kind of touched on that earlier when we were talking about specific binding of the substrate to a specific enzyme, right? That's the whole basis of this. If you write it out such as this, enzyme plus substrate forms a enzyme substrate complex, giving you an enzyme and product. This is the simple formation of that. And then you can draw a picture to go along with it that might explain it. So you have your enzyme plus your substrate goes to enzyme substrate complex.
So the enzyme plus substrate gives you a complex,
Enzymes.
It does give you a complex.
giving me a
Enzyme
complex.
plus substrate forms a,
Enzyme, substrate?
So,
you can start over.
if I was to draw it out in picture form, you have your enzyme plus your substrate, enzyme substrate complex, enzyme product. Enzyme plus your substrate, enzyme substrate complex, enzyme product, right? But you want to explain what that means, right?
I can see.
You want to explain what that means, right? What that means is enzymes will only bind to substrates that fit specifically in that active site, right? Once they fit, they form a complex. Now, that enzyme has the ability to work on that substrate. Doesn't matter whether it is a hydrolysis reaction or a dehydration reaction, because enzymes can do both of those, right? But we have this complex that is formed. After that substrate has been worked upon, you're going to get a product, whether it be breaking apart or building together, okay? But you will get a product at the end of it. So here's your lock and your key. When the key fits specifically into that lock, it will open up that door, right? Giving you